Hey, welcome back. Mr. P here. This is the third lecture of chapter 10 already out of 4 total lectures. Getting closer to the end. And in this lecture, we're going to talk specifically about mitosis. Last lecture, we talked about the states of DNA, both chromatin and chromosomes. We talked about what binary fission is and what has to happen to the DNA in order for it to become supercoiled and condensed into all of the structures described, which were chromatin condensing to form 2 sister chromatids held together by a centromere. Both of those chromatids held together by a centromere produce an X-shaped chromosome, and that centromere that holds them together is essentially going to be the thing or the structure that will attach the spindle fiber between it and the centriole in order for that chromosome to be moved around. But let's move into what mitosis is and the nuts and bolts of the cell cycle. And so I'd like to start with the first meme of chapter 12, I believe. It's been a while, and my students have been starting to pester me a bit about where they went and why they aren't still in. And so we have a chromosome, and we have chromatin, and the chromosome is mad and says, dude, mitosis starts in 5 minutes. I can't believe you're not condensed yet. And he just looks shocked, like, how is he going to condense in 5 minutes and be ready for mitosis? But alas, he will, and this chromosome or mess of chromatin will condense into an X-shaped chromosome. And so we can see here that this would be the centromere, this would be a sister chromatid, this would be a sister chromatid, and they don't typically have eyes and a mouth. But that's what makes it funny. So let's talk about a little bit related to the cell cycle. Okay, so the cell cycle consists of phases and subphases, and there are 2 main phases of the cell cycle. The cell cycle consists of interphase and mitosis. Interphase consists of G1, S, and G2. Okay, G1, S, G2. Mitosis, like I've talked in class, is made up of P, M, A, and T. Prophase, metaphase, anaphase, telophase. So we have gap 1, synthesis, gap 2, prophase, metaphase, anaphase, telophase, and then we have the last cytokinesis event, which we've talked about before. And cytokinesis, remember, is the division of the cytoplasm. Mitosis specifically is the division of the nucleus, and interphase is when the cell does all of its normal cellular things. Okay, so here's the cell cycle. Everything in this kind of orangey, peachy color is interphase, and interphase is the main portion of that cell's life. The only thing that happens in this red and blue area of this entire pie chart is cell division. Okay, so mitosis is the red part, P-M-A-T, PMAT, in this direction. Okay, PMAT. And the blue, cytokinesis, is just the division of the cytoplasm. Okay, and so the cytoplasm must divide, the nucleus must divide. All of this constitutes what we call as cell division. And all of this, in this orangey kind of peachy color, is interphase. Now, I said in the previous slide that interphase is made of G1, S, and G2. G1 stands for gap 1. It is the main phase of interphase, or the longest phase of interphase, and this phase is when the cell grows. Okay, so the cell is going to physically get bigger. It's going to start small, and it's going to grow to a larger size during gap 1. Okay, so this cell is going to grow. It's also going to replicate organelles, and you might think, why does it need to replicate the organelles? And the answer is, we are eventually going to divide this cell from 1 into 2, and so we need to replicate all the organelles so that each new daughter cell has a complete set of organelles. If you don't replicate the organelles and then you divide all the organelles, each cell is going to have half the organelles they really need. For instance, if you don't replicate or make more rough ER, then one cell is going to get the rough ER and the other cell isn't going to have any rough ER because there's only one rough ER. So we need to duplicate the rough ER, we need to duplicate the organelles so that each new daughter cell has a full set of organelles. In S phase, S stands for synthesis. And synthesis means basically to copy or to build, and so we are going to replicate our DNA at this point. What does that mean? We're not going to grow, we're not going to divide organelles, we're not going to replicate organelles, we're just going to copy our DNA. And so one double helix is going to produce two individual double helices. Okay, so one DNA is going to copy itself and it's going to become two. We're going to learn in chapter 12 how the DNA replicates itself, but for now just know that one DNA molecule is going to make two DNA molecules, and that's so that one cell gets one set and the other cell gets the other set. And then in gap 2, which is the same as G1, you just have a little additional growth, and this is a very short process, but it's important because the cell needs to grow a little more in order to become the entire size, or become the size needed to eventually divide and be two viable, mature daughter cells. Okay, so interphase is G1, S, and G2. Mitosis is composed of prophase, metaphase, anaphase, telophase, and the very last is cytokinesis. Cytokinesis is the division of just the cytoplasm. Interphase, the first part of the cell cycle. Interphase, and if I had a little song for anyway. Cell cycle, multiply cells. Whoa! That one got away from me. Interphase is made up of three subphases. Remember, I said G1, S, and G2. This is just kind of going through each of those subphases. The first subphase is G1. It's the first growth phase, but remember G stands for gap. And so even though growth starts with a G, it's a good way to remember that growth occurs in G1 and in G2, but G stands for gap. And so gap 1 is the first growth phase. Cell grows in size, it gets larger, and increases the number of organelles so that each new daughter cell will have a complete set of organelles. If we go into the second phase of interphase, that is S phase, and S stands for synthesis. And in the S phase, or synthesis phase, DNA is going to duplicate itself. Okay, so DNA is going to be duplicated, and one distinction to make, or one kind of side note to make, is that DNA in the S phase is still in the chromatin state. And remember, the chromatin is the long, stringy strands of DNA, kind of like the meme I showed you at the very beginning, where it wasn't condensed. Okay, chromosomes are still in this state during S phase. Okay, still in this phase, or still in this state during S phase. And it's not going to be until M phase of mitosis where the chromosomes are completely condensed. They will start condensing in prophase and start moving to the center, but they're typically not in M phase, or not in chromatin or chromosome state until M phase. Okay. The third subphase is G2. G2 is the second growth phase, and cell grows in size, and again, it's the last ditch effort to increase the number of organelles to the levels needed in order to divide. Okay, so interphase, G1, S, G2. G1 and G2 are your gap phases, and they are your first and second growth phase. They're also going to increase the number of organelles in order to divide the cell. S phase, you duplicate the DNA. Mitosis. Today we're going to learn about mitosis. I need your undivided attention. Get it? Because prior to mitosis, you are undivided. Anywho, the cell cycle multiplies cells. In mitosis, there is a... so mitosis, first of all, is the second main phase of the cell cycle. Remember, the first main phase is interphase. The second main phase is mitosis. And mitosis is the shortest of those two phases. But of the time spent in mitosis, there are 4 subphases. And so all 4 of these would be basically happening at different times in this order, first, second, third, and fourth, that make up mitosis. So prophase is the first phase or subphase, metaphase is the second subphase, anaphase is the third subphase, and telophase is the fourth subphase. Here is a cell in prophase. Okay, and so the first subphase of mitosis is prophase. Remember PMAT. In prophase, there are several things that have to happen in order. The first of which, the centrioles, which are these guys, start their migration. They would have started together. They are moving to the poles of the cell so that they can basically take up residence here and here. They want to be completely on opposite sides of the cell from each other so that they can basically orchestrate the division of this nucleus. Okay, so they migrate to the poles. Spindle fibers begin to form. All of these little fibers right here are forming off of the centrioles. These centrioles are that structure. The spindle fibers are these structures. And so not only are we going to have spindle fibers between these two centrioles, but we're also going to have spindle fibers that are eventually going to attach to the centromere of each of the chromosomes. Okay, that's what's going to eventually move these chromosomes around. Nuclear membrane begins to break down. See how this nuclear membrane now has these sections that are completely dissolved, and this nuclear envelope, which is normally around the DNA, specifically around the chromatin during interphase, is designed to keep the chromosomes safe, but we can't divide the nucleus while this really protective wall or membrane is around the chromosomes, and so we want to break that down. That would be here. And chromatin condenses into chromosomes. Remember, our chromatin is our long stringy strand of DNA. It's going to condense into the chromosome structure, which is that structure that is X-shaped, held together by a centromere. And you can see that these chromosomes are starting to form. We have that X-shaped. So that's prophase. Here is interphase. Again, our DNA is in the form of chromatin. Our centrioles are together. Now, a pair of centrioles with all of the components necessary to divide these up is called a centrosome, but we don't necessarily need to know that. Just know that these are centrioles. Chromatin is in the nuclear envelope with very small nuclear pores in the nucleolus contained within this nucleus region. And so during prophase, we are going to not only start dissolving this nuclear membrane or this nuclear envelope, we're going to start moving these centrioles or centrosomes to the poles. We're going to produce spindle fibers. We're going to start condensing this stringy strands of chromatin into long kind of X-shaped chromosomes, which contain two sister chromatids each held together by a centromere. Eventually, at the end of prophase, notice that this is the beginning, early prophase, and this is kind of late prophase. In late prophase, our nuclear envelope is basically completely dissolved. It is no longer, the chromosomes are no longer contained within the nucleus. These kinetochores are specific structures or specific proteins within the centromere that allows the spindle fibers to attach to that centromere. And so these have all attached themselves. The centrioles have made it to the poles, and they are eventually kind of on their way to being lined up along this middle plate. Metaphase, PMAT, prophase, metaphase. Metaphase is our second phase in metaphase. It's the easiest one to recognize because the centrioles have ended their migration. Notice they are at the poles. One is on this side, one is on this side. The spindle apparatus is completely set, meaning all of the chromosomes are attached to the centrioles via the spindle fibers. And the chromosomes are lined up in the middle. This is called the metaphase plate. Right along the equator of this cell, the chromosomes have lined up. And when you look at a cell and notice that the chromosomes have lined up in a line, it's a pretty good indication that the cell is in mitosis, specifically in metaphase of mitosis. Anaphase, the third phase of mitosis. We are going to start pulling the chromosomes apart. So anaphase starts with an A. Apart starts with A. Another thing I forgot to say back here when we were talking about mitosis, specifically metaphase, metaphase starts with M, so does middle. Okay, so if you think about the chromosomes lining up in the middle, that's metaphase. Metaphase is middle. Anaphase, A, and apart. So centromeres are going to release. Remember, the centromere is the point at which these sister chromatids are holding each other. And so because the centromeres release each other, the sister chromatids are separated and become individual unduplicated chromosomes. And because they have been pulled apart, they are going to be pulled towards the poles, towards the centriole, so that eventually we have a lot of room right here in the middle to divide the cytoplasm during cytokinesis. And telophase, the fourth phase or the fourth subphase of mitosis. Telophase, it begins with a development of a cleavage furrow. The cleavage furrow is right here. It's where the cell is pinching in. Okay, it forms a cleavage furrow. This is ultimately going to be where cytokinesis takes place. The further it pinches in towards each other, the closer it is to actually dividing the cytoplasm. And so the cleavage furrow divides the cell or starts pinching in. Nuclear envelope begins to reappear. Notice that the nuclear envelope is starting to reappear around each of these unduplicated chromosomes. Chromosomes begin to decondense back into the long stringy strands of DNA called chromatin, and eventually cytokinesis occurs right here within that cleavage furrow. If we look at these, again, metaphase, middle, and so the chromosomes have lined up in the middle. The centrioles have ended their migration. All of the spindle apparatus is set. This middle equator is the metaphase plate. And as soon as they line up in the middle, they're going to be pulled apart right here at that metaphase plate. And remember, anaphase, apart. We are splitting the chromatids apart at the centromere. And so remember, each of these chromatids should be identical. And because these are identicals, each of these new daughter cells should be identical. And so those are going to basically be pulled back towards the poles. And once you separate these chromatids at the centromere, we develop this cleavage furrow, which is where the cell membrane starts pinching in. This is where cytokinesis will take place. The nuclear membrane starts forming again around the chromosomes. These chromosomes start decondensing back into the long stringy strands of chromatin, and our spindle fibers start dissolving, and basically these centrioles kind of get ready to begin the next migration when it's ready to go through cell division again. So we talked about this idea of genetic continuity in the first of lecture 10, okay, or the first lecture within chapter 10. And mitosis, remember, ensures genetic continuity from generation to generation because we are making identical copies of the DNA before splitting the cytoplasm. Cells that go through mitosis begin as a diploid cell and they end as a diploid cell. Remember I said diploid is 2N. And so we are starting, for instance, a skin cell has 46 total chromosomes. After duplicating each of the 46 chromosomes and dividing into 2 new cells during metaphase, each new phase or each new cell will have 46 total chromosomes each. And that's easy to believe because a skin cell that is part of your skin tissue should become 2 identical skin cells that each have the same amount of chromosomes each. Okay, so that's a diploid cell becoming 2 new diploid cells. What is the difference between plant and animal cell division? Well, we've talked about animal cell division so far, and so if you think about animal cell division during telophase, remember it's going to produce that cleavage furrow, and eventually it's going to pinch in close enough. If I keep going, say this is a time lapse, it's going to keep pinching in closer and closer and closer until eventually it splits. Well, this cytokinesis can't happen the same way in a plant cell because plant cells have really thick cell walls. These cell walls that extend around the whole outside of the cell membrane are too rigid to pinch in like an animal cell does. And so the way in which a plant cell goes about dividing the nucleus is the same. So plant cells still go through the same mitosis events because we want to divide the nucleus. But then it has to start forming this cell plate, which means we're going to form a new cell wall between the two new daughter cells. Okay, we can't pinch a plant cell in because of the cell wall. We have to just build a new cell wall between the two new daughter cells. Okay, so that's the difference between animal cells and plant cells. Have you ever wondered why it's called cell division when the numbers multiply? We keep talking about one cell becoming 2, and 2 cells becoming 4, and 4 cells becoming 8, and 8 cells becoming 16, and 16 becoming 32. So why are we multiplying when it's called cell division? I don't know. You tell me. Anywho, this joke has nothing to do with mitosis, but I thought it was pretty funny. And so, how are a dog and a marine biologist alike? One wags a tail and the other tags a whale. It's up for you to decide which is which, but until next time, see ya.